The paper that's described in the second of the cancer lectures really asks the question, firstly, if we sequence lots of different cancers, which is what they did, they sequenced 21 different cancers from several thousand individuals. They asked the question, can we find evidence of all of these pathways across all of these cancers? Are there any other pathways? And are there genes which are mutated in many different cancers, is what they're asking. So the first thing they found is that there's a 11th hallmark of cancer, which is epigenetic and RNA dysregulation. This is all about genes and mutations that change chromatin, that change gene expression. It doesn't do this through targeting enhancers, but it does this through targeting the proteins that bind to enhancers, what we call chromatin remodeling enzymes, of which the protein SMARCA4 is an example. So this is a diagram. Again, it perhaps looks complicated at first look, but it actually has a lot of information in it. And I would strongly suggest everyone to take time to really think about the information in these kind of slides. So first of all, this circle with all these symbols on it is explained down here. Each little symbol represents a different hallmark of cancer, whether it's cell death, shown as the little gravestone cross up here, or whether it's, for example, genome instability and increasing mutations, shown with this particular symbol here. So what they're showing here is this is, through sequencing a range of different cancers, what % of cancers showed mutations in these particular pathways. And you can see there are some that are fairly common. 55% of all cancers sequenced show mutations in these proliferative signaling pathways. And we can see the KRAS, BRAF that we've just been talking about. These are typically the receptor tyrosine kinases. We can also see this new hallmark, which is epigenetic regulation, and that includes the gene SMARCA4. And this is what's actually regulating chromatin structure and enhancer activity. This is a result from the sequencing of all of these many different cancers. And again, there's lots of information on this slide, but we can simplify it by just looking at what it's telling us. So first of all, we've got the tumor types, and there's a range of different tumor types. There's the number of normal and tumor pairs. So in other words, if you're going to sequence a tumor, you need to sequence a bit of healthy tissue from that person as well. Most cancers are sporadic, which means that a mutation will only occur in that cancer cell and not in any other somatic cell in the body. So as I said earlier, whole genome sequencing gives us an encyclopedia of common and rare variants in healthy cells. So by sequencing both healthy and tumor cells, we can identify mutations that are associated with cancer. Different cancers have different numbers of mutations. So for example, breast cancer, 32 genes, endometrial cancer, 58 genes show mutation, and there are others, rhabdoid tumors, just require a single mutation in order to become a cancer. The rhabdoid tumor is actually a very nasty early childhood developmental cancer. And it's a mutation typically in a gene related to SMARCA4 called SMARCA2. So it's a gene that disrupts early patterns of epigenetic modification of genes in the developing embryo. And it's a very early childhood cancer. And it just requires that one mutation to lead to these cancers. So it's a bit of an exception. Most cancers have many genes that are mutated. This is SMARCA4, and SMARCA4 is an ATP-dependent chromatin remodeling protein, and it is made, and there are a range of different mutations, truncating mutations. So this might be a nonsense mutation, for example, and there are many missense mutations. And you can see that SMARCA4 is mutated in many different types of cancers: kidney, endometrial, bladder, stomach, lung, and so on. And the mutations tend to be centered in the active domain, which is an ATPase domain. So this study that I've I described in the online lectures, what they were asking was, first of all, the hallmarks of cancer, and they clearly found evidence for these chromatin remodelers. But secondly, they looked for mutations that were found across a range of different cancers, and SMARCA4 is one of those mutations. Because in cancer therapy and diagnosis, we want to be able to understand not only what differentiates different cancers, but also what is common to many different cancers. And of course, that would have implications for treatment and so on. That's it for, I think, for this. Oh no, there was one more slide. Yes, so SMARCA4 is all about chromatin remodeling. And what that means is that we go from this highly compact chromatin structure that we see here, that the remodeling enzymes or the remodeling complexes, because SMARCA4 is actually part of a very large remodeling complex, what it does is it starts shifting the nucleosomes, it starts opening up stretches of DNA and allowing access of transcription factors. And at the same time, we start to see these histone acetyl transferases start to weaken the interaction between the histone and the DNA as well. And this means that the promoters and enhancers can now bind transcription factors and transcription can proceed. It's thought that the loss of SMARCA4, what it does is it turns genes on. It's a remodeling, part of a remodeling complex called SWI/SNF. And what it does is that it prevents inhibition by another complex, which is called PRC, which is the polycomb repressor complex. And importantly, it controls the balance between differentiation and self-renewal of cells. So if we go back to our acute leukemia, for example, we saw that initiated in the stem cell compartment of the bone marrow. And it's increasingly being seen that many cancers are thought to arise from stem cells. And that there is a lot of work being given to the identification of what are called cancer stem cells. Because a cancer cell really is a highly proliferative, poorly differentiated cell. And a stem cell is a cell that can keep proliferating and it is not differentiated. So cancer cells and stem cells share some similarities. And it's thought that in a number of cases, it is mutations in stem cells which keeps those cells as being types of stem cells, but of course removes all those normal checks and balances to make sure that they don't become malignant and metastasize. And it's thought that SMARCA4 plays a very important role in this, in that in the absence of SMARCA4, which is shown down here, we see a loss of differentiation and an increase in self-renewal. So the loss of SMARCA4 upsets the balance between maintaining a differentiated, non-renewing somatic cell, such as a lung cell or a muscle cell or a liver cell, and instead it allows the appearance of these cancer cells, which tend to be less differentiated and highly proliferative. So that is one of the key findings from these whole exome studies, the discovery of a very large class of chromatin remodeling enzymes and their effect on cell identity, and particularly raising these questions about what is a cancer cell and its relationship, for example, to stem cells. Okay, I think that's all of the slides. The other questions here were about the enhancer and epigenetic modifications, that we see that enhancers typically acquire marks such as lysine acetylation and histone lysine 4, methylation, which is a mark of an active gene, and the epidermal growth factor receptor is a receptor tyrosine kinase that is often mutated in lung cancer and a type of brain cancer called glioblastoma, which is a very, very nasty cancer, and that these mutations in EGFR result in EGFR being on. It no longer needs its ligand, which is called epidermal growth factor. And you see this tyrosine kinase pathway starting to change patterns of gene expression to remodel the way genes are turned on and off. So what we've seen is that Sequencing of cancer genomes is incredibly important because it can map groups of genes which tend to be mutated in a range of different cancers. It also tells us that an important basis of cancer is the remodeling of the chromatin across our genome, whether that's very early in life or whether it's later in life in these somatic cancers. But at any rate, what it does is it tends to favor proliferation and de-differentiation.